Welcome to this lecture on human fertilization. We'll begin with a short 3D look at the journey from sperm entry to the formation of the zygote and early blastocyst. By the end, you'll have a solid understanding of how fertilization begins and how early development unfolds. The female reproductive system is located in the pelvic cavity, nestled within the pelvic bones. It includes internal organs such as the ovaries, fallopian tubes, uterus, cervix, and vagina, as well as the external genitalia, vulva. Ejaculation and sperm entry. Around 100 to 300 million sperm are ejaculated into the vagina, beginning their journey toward the oocyte. Most sperm die in the acidic vaginal environment, and only a small fraction survive to reach the cervix. Passage through the cervix. Only sperm passing through ovulatory thin cervical mucus enter the cervix and continue toward the uterus. Movement through the uterus. Uterine muscular contractions help transport surviving sperm upward, further reducing their numbers. Few sperm reach the oviduct. Only a few hundred sperm reach the fallopian tube isthmus, acting as a reservoir near the ampulla. Capacitation, 5 to 7 hours. Sperm undergo capacitation for approximately equals 5 to 7 hours, improving motility and preparing their membrane to penetrate the oocyte layers. The ovulated secondary oocyte remains viable for only 12 to 24 hours, making the timing of fertilization critical. During this window, the oocyte awaits the arrival of sperm in the fallopian tube. Sperm-oocyte encounter. Capacitated sperm reach the oocyte in the ampulla and begin interacting with the corona radiata and zona pellucida. Penetration and fusion. One sperm penetrates the zona pellucida and fuses with the oocyte membrane, initiating the fertilization event. Block to polyspermy. The oocyte triggers a cortical reaction that prevents additional sperm from entering, ensuring only one paternal genome is included. Completion of meiosis II. Following fusion, the oocyte completes meiosis II, forming the female pronucleus while the sperm forms the male pronucleus. Pronuclear preparation for mitosis. The male and female pronuclei migrate, replicate DNA, and align for the first mitotic division. A solid ball of 16 to 32 cells, called the morula, forms by day 3 to 4 and continues its journey through fallopian tube towards the uterus. By day 5, the blastocyst, containing an inner cell mass and fluid-filled cavity. The blastocyst attaches to the endometrium, starting implantation around day 6 to 7. This diagram highlights the major steps of fertilization and early embryonic development. It shows how the sperm fuses with the oocyte, activates it, and triggers protective reactions. The male and female pronuclei then form, migrate, and unite, leading to the first mitotic division. Finally, the embryo begins its journey through cleavage, morula, and blastocyst stages toward implantation. Step-by-step mechanistic sequence. Step number 1: ejaculation and deposition in the vagina. During ejaculation, 100 to 300 million sperm are released into the vagina. In vagina, the acidic pH, approximately equals 3.5 to 4.5, and innate immune defenses rapidly destroy most sperm. Seminal plasma contains bicarbonates and other buffers that neutralize this acidity, but its proteins must later be removed during capacitation. Step number 2. During ovulation, cervical mucus becomes less viscous, oestrogenic effect, forming channels that allow motile sperm to ascend. Only the most motile, morphologically competent sperm navigate these channels. Step number 3. Uterine transport. Uterine contractions, myometrial peristalsis, Ciliary activity and fluid currents assist sperm transport toward the oviducts. Seminal plasma is largely left behind. Sperm are capacitated while within the female tract. Step number 4. In the isthmic portion of the fallopian tube, sperm attach transiently to oviductal epithelium, a reservoir. This prolongs their survival and times release to coincide with ovulation. This interaction also contributes to final maturation changes. Step number 5. Capacitation, sperm activation. During capacitation, some cholesterol is removed from the sperm membrane, which makes the surface more flexible. At the same time, bicarbonate and calcium ions enter the sperm through specific channels. These ions activate adenylate cyclase, which raises CAMP levels inside the cell. The rise in CAMP switches on protein signals that boost tail power and produce hyperactivated motility, helping the sperm push through cumulus cells and move toward the zona pellucida. Step number 6. Ovulation and the structure of the released oocyte. During ovulation, the ovary releases a secondary oocyte, which is paused in metaphase 2 of meiosis. The egg is not alone. It is surrounded by 2 important protective layers. I.e. the corona radiata, cumulus cells. A cluster of granulosa cells held together in a soft, jelly-like hyaluronic acid matrix. Zona pellucida. ZP. A thick, transparent glycoprotein shell made of ZP1, ZP4 proteins that protects the oocyte and helps with sperm recognition. The oocyte can only be fertilized for a short window of about 12 to 24 hours after ovulation. Step number 7. Migration to the ampulla, which is site of fertilization. Fertilization typically occurs in the ampulla, the wider, more distal region of the tube where sperm and oocyte meet. Step number 8. Penetration of the cumulus or corona radiata. Capacitated sperm penetrate the cumulus matrix using both mechanical force, hyperactivation, and enzymatic activity, for example, hyaluronidase, that degrades hyaluronic acid, dispersing cumulus cells. Step number 9. Binding to the zona pellucida, species recognition. Sperm bind to the zona via specific interactions between sperm surface receptors and zona glycoproteins, ZP proteins. In humans, ZP3 and ZP4 are implicated in initial binding and triggering events. Subsequent binding and passage involve ZP2 and structural. ZP1. The ZP's glycoproteins bear O and N-linked oligosaccharides that are important for the ligand receptor specificity. Step number 10. Acrosome reaction. Enzyme release for zona penetration. The acrosome is a cap-like vesicle on the sperm head, filled with enzymes. When a capacitated sperm binds correctly to the zona pellucida, especially ZP proteins, its membranes fuse and the acrosome releases these enzymes. Released enzymes, such as acrosin and N-acetylglucosaminidase, begin digesting pathways through the zona pellucida, allowing the sperm to move forward. This reaction also exposes new proteins on the sperm surface, on the inner acrosomal membrane, that are needed for the next binding step, for example, binding to ZP2. Only capacitated sperm can undergo a proper acrosome reaction. Without it, the sperm cannot penetrate the zona pellucida or proceed to fertilization. Step number 11. Zona penetration and entry into perivitelline space. Acrosome-reacted, hyperactivated sperm bore a path through the ZP by combined enzymatic digestion and mechanical thrust. Entry into the perivitelline space places the sperm adjacent to the ulema, oocyte plasma membrane. Step number 12. Sperm-oocyte membrane recognition and fusion, ulema fusion. Membrane fusion is mediated by specific molecular pairs. In mammals, Izuma-1, sperm surface, binds to Juno, oocyte receptor. This is essential for membrane fusion. Additional sperm proteins, for example, pH30 fertilin components, and oocyte microvillar architecture localize and facilitate fusion. Upon fusion, the sperm tail or flagellum typically ceases beating. The sperm plasma membrane and oocyte microvilli interact, and the sperm head, midpiece, and associated structures are drawn into the oocyte cytoplasm while the tail is left behind or degenerates. Step number 13, oocyte activation. C² oscillations and downstream events. Sperm entry introduces PLC into the oocyte, triggering PP hydrolysis and IP production. IP binds to A receptors, releasing Ca² and generating cytoplasmic Ca² waves. These calcium oscillations activate meiosis completion, cortical granule exocytosis, and metabolism. The cascade ensures successful egg activation and blocks polyspermy. Step number 14, cortical reaction, which help to block polysperm's entry into the egg. Cortical granules fuse with the eulemma and release contents, proteases, mucopolysaccharides, peroxidases, into the perivitelline space. These biochemical modifications cause zona hardening and destroy sperm-binding receptors, for example, by cleavage or chemical modification. Step number 15, male pronucleus formation as the sperm decondensation. After the sperm enters the oocyte, its tightly packed chromatin unwinds, replacing protamines with maternal histones, and the nucleus re-expands to form the male pronucleus. The sperm brings more than DNA. It usually provides the centriole centrosome, which is crucial for assembling the first mitotic spindle of the embryo. It also delivers small amounts of cytoplasm, proteins, and regulatory RNAs that can influence early embryonic development. Although the sperm enters with mitochondria, these paternal mitochondria are actively destroyed, ensuring that the embryo inherits mitochondria almost entirely from the mother. Step number 16. Female pronucleus and pronuclear apposition, syngamy. After completing meiosis II, the oocyte forms the female pronucleus, while the male pronucleus is forming nearby. Both pronuclei move toward each other, guided by microtubules organized by the sperm-derived centrosome. They stay separate but side by side, each replicating its DNA. Their membranes break down just before the first mitosis. Syngamy actually occurs when maternal and paternal chromosomes align together on the first mitotic spindle, creating the first diploid nucleus of the embryo. Step number 17. First mitotic division and zygote formation. The first mitotic cleavage produces 2 blastomeres. The embryo is now a zygote and development proceeds through cleavage stages 2, 4, 8 cells, compaction, approximately 8 cells, morula, and blastocyst. Step number 18. Embryo transport and implantation timing. The developing embryo travels down the tube over 3 to 5 days, forming a blastocyst by day 5 to 6, which then implants into the uterine endometrium around day 6 to 7 post-fertilization. Thank you for watching. If you found it helpful, please like, subscribe, and share to support more lectures like this.